Hey everyone, it's Dr. Markan, and we are going over Chapter 7 in your book, The Cellular Basis of Inheritance. So basically, we're covering the other cell division, which is meiosis. Some of the questions that we're going to cover include, how are our sex cells made, or our gametes? Why are you so genetically unique? And then what happens when things go wrong in meiosis? So some of the guiding questions in this chapter include, how does the cell mix up the DNA from each parent, which gives us that genetic variation? And then how does the cell split the mixed DNA in half? We know that the DNA during meiosis will replicate once and then divide twice. So that kind of gives you an idea of how we are able to split that DNA. So some of the videos that could help you with this topic include the video on meiosis by the Amoeba sisters, and here's that link, as well as another video on meiosis, and that link is located there. For our learning objectives for this chapter, you should be able to identify or describe the key phases of meiosis and how they are similar to and different from mitosis. So kind of compare and contrast between meiosis and mitosis. Describe how gametes, or the sex cells, are made in humans and why siblings can look so different from each other. So part of your discussion question this week, you run into your old friend Manuel, who tells you that, strange as it might sound, he and his identical twin brother, Jose, got married to identical twin sisters. What are the chances? He says it's going to be really crazy because both couples are now pregnant. So his and Jose's kids will also be identical twins. Let's think about that. So this week's discussion question is, using your understanding of how sperm and ova, or the egg, are made, what do you say to Manuel in response? And of course, you have to provide at least 3 pieces of evidence to support your response. And here are actually some articles to help you answer this discussion question. So are their babies, their individual babies, going to be identical? So let's take a look at the processes of meiosis to see whether or not this is more than likely or not more than likely. Here we have the human life cycle. We have a man and woman coming together, donating an egg from the mom, sperm from the dad, to cause fertilization. With fertilization, we have a fetus that will eventually become their child. So with fertilization of the egg, this egg, we will say, is diploid in number, meaning it has 46 chromosomes. So diploid 2n equals 46. And of course, this fetus and the cells within the fetus, or the body cells of this fetus, undergoes multiple divisions, multiple cycles of mitosis to form this human organism. And again, the process of mitosis continues, allows this baby to grow up and eventually be able to reproduce and have children of their own. So what is meiosis? Meiosis, we know, is a specialized process that produces a male gamete, sperm cell, or a female gamete, which is an egg cell. So meiosis deals mainly with our gametes, or sex cells, whereas, as discussed previously, mitosis is the process that involves your body cells, or your somatic cells. Okay, so that's definitely a huge difference between meiosis and mitosis. We know that the gametes from a male and a female are haploid in number, meaning the egg will have 23 chromosomes, and the sperm will also have 23 chromosomes. So they are haploid in number. Okay. So once we have fertilization of these 2 gametes, we will then produce a cell that is diploid in number. So meiosis in males occurs in the testes, which are the reproductive organs of the male reproductive system. Meiosis takes about a month for the entire process to occur in males. And this process will produce several million sperm per day. In females, meiosis occurs in the ovaries. The ovaries, of course, being the reproductive organs of the female reproductive system. Prophase I of meiosis begins before birth, usually about 5 months age of gestation. And then the meiotic process will end decades later during the monthly ovarian cycle, and females will produce 1 egg per month. So prophase I is paused until the female reaches puberty. So in males, gametogenesis, which is the formation of the gamete or the sperm, will begin at puberty, whereas in females, gametogenesis actually begins 5 months age of gestation, which allows for differentiation or prophase I of meiosis to the primary oocytes. So you, as an individual, as an organism, will inherit half of your chromosomes from mom and half of your chromosomes from dad. So normally, the egg will have 23 chromosomes. Dad will contribute 23 chromosomes in the sperm, and when we have fertilization, the normal embryo will have 46 chromosomes. So these chromosomes, 22 chromosomes from mom and 1 X chromosome, and then 22 from dad, and then either an X or Y chromosomes. So in order to be physically identified as a girl, the sex chromosome will be XX, and to be identified as a boy, the chromosome would be XY. Okay? So it's a little bit more complicated. Mom actually contributes slightly more than half of the DNA to the baby because the mother provides all mitochondria for the fertilized egg. But just to keep things simple, we're going to say, mom contributes 22 chromosomes plus an X chromosome, as well as mitochondrial DNA, and then dad 22 chromosomes and either an X or Y chromosome. Okay, so a total of 44 chromosomes plus another chromosome, the sex chromosomes from both mom and dad, either XX or XY, plus mitochondrial DNA coming from mom. And here is another YouTube video. This link is actually a funny little video called a Biologist's Mother's Day song, talking about the contributions of chromosomes as well as mitochondrial DNA from mom. So if you want a song stuck in your head, definitely go follow this YouTube link. So now in the process of meiosis, cells will split from one cell to 4 cells. So the end product is basically 4 cells. So every new cell has half the amount of DNA as the parent. So here we have the original cell, which will have 1 round of replication. Then the cell will divide not once, but twice to create 4 cells. Now, these 4 cells are not identical to the original cell. So here we see the male gametes, the sperm cells, and each one is actually different than the original parent cell. Again, not identical to the original cell. And these cells are considered haploid, so 23 chromosomes. Now recall in mitosis, every new cell is the exact copy, or has the exact copy of all its DNA from the parent cell. So DNA replicates and then divides. And we have two daughter cells that are completely identical to the parent cell. So the new cells, the daughter cells, are identical to the parent cell. That is mitosis. That's another way that mitosis is different from meiosis. So meiosis also promotes genetic variability, makes us all different, makes us look different. So how does the cell mix up the DNA from each parent, providing that genetic variability? We know that the DNA you inherit from each parent actually gets mixed up as the gametes are made. So in your parents' gonads, or reproductive organs, their homologous chromosomes are paired up and exchange DNA. So this is what's known as crossing over. So here we can see the homologous chromosomes pairing up, and then they cross over to exchange DNA. Crossing over. So when the sperm or ovum is finally made, each cell didn't get an exact half of the original. It actually got a mixed half. So maybe another way we can kind of discredit what Manuel is saying about having identical twins. So crossing over, one piece of evidence to show that perhaps their kids aren't exactly that identical. Another way that ensures genetic variability is something called independent assortment. So when your parents make sperm or ova, the chromosomes, after crossing over, will then get randomly combined into gametes. So we have random combinations of chromosomes that will either become, or that will be placed into either sperm or egg's cell. So with independent assortment, first we'll have our chromosomes lining up, and then we know that the red stands for maternal derived chromosomes, and the blue standing for or representing the paternal or father derived chromosomes. And then after an independent assortment of these chromosomes, we can see that they are all mixed up. So we might have paternal derived chromosomes on one side, or maternal derived chromosomes on another side. So note, some blue and some red on one side. So the chances these would line up the same exact way every time the gametes are made is extremely rare. So independent assortment, again, another way that we have genetic variability. So in humans, we know that N is equal to 23 chromosomes. And then we have, if we do the math, we can actually have 8 million combinations of the pairs of chromosomes. So again, another piece of evidence to show that maybe Manuel and Jose's kids will not be identical twins. So choosing the same chromosome sets for siblings would be extremely unlikely. So the chances of getting this exact combination from mom and dad to make you is roughly 1 in 70,368,744,177,664. So you're saying there's a chance. But still, it is very highly unlikely that you will get the exact combination from mom and dad to make you. So if we take a look at this picture is actually called a karyotype. A karyotype is an actual representation or image of chromosomes. So here we have circled your dad's 23 chromosome pairs. So these circled chromosomes go into the sperm. Here we see the karyotype from mom, that's showing mom's 23 chromosome pairs. So the circled chromosomes are what's going into the egg. And when we have a combination of the egg and the sperm, we get U. So for this to happen twice for identical siblings is highly unlikely. So let's consider something else called random fusion. So from hundreds of millions of sperm, all with different combinations of DNA, only one random sperm will fuse with the egg. Okay, and here we have our winner. Fusion of two gametes, an egg and a sperm, form a zygote. So each gamete has about, like we said, 8 million possible chromosome combinations due to independent assortment. This gives us over 70 trillion diploid combinations. Okay, so again, it is very highly unlikely that you are getting the same exact chromosomes from mom and dad. So now let's talk about the actual process of meiosis. How does the cell split the mixed DNA in half? Again, we have to remember that in meiosis, DNA will replicate once and divide twice. So how to get haploid cells? Again, chromosomes will replicate once but divide twice. And remember, meiosis occurs within the testes and ovary to produce male gametes or sperms in spermatogenesis and a female gamete or egg during oogenesis. So during meiosis I, we have separation of our homologous chromosomes. So we go from a diploid number to a haploid number, so 2N to N. And during meiosis II, the sister chromatids will separate just like in meiosis, and then we have go from a haploid situation to another haploid situation. So here we see the basic breakdown of meiosis. First, we have the interphase of meiosis I. Here we have the homologous pair of chromosomes in a diploid parent cell. We then have replication of our homologous pair of chromosomes, creating sister chromatids. At this point, the cell is diploid with the replicated chromosomes. And then we have our various stages where we have crossing over, independent assortment. Essentially, in meiosis I, first we'll have our separation of the homologous chromosomes, and eventually we'll have the haploid cells with replicated chromosomes, at which point we enter meiosis II, where we'll have another division. So the sister chromatids will separate, and here we have our 4 haploid cells with our unreplicated chromosomes. So again, we only have one replication that occurs in meiosis, but 2 divisions. So during meiosis I, the homologous chromosomes will separate. So we go from 2N, which is diploid, to a haploid situation. So the diploid number to haploid number, where a primary spermatocyte in males and a primary oocyte in females will become either a secondary spermatocyte or a secondary oocyte. Now, it's important to note that in females, eventually, when we have the end product, only one egg will be chosen per month. So with those 4 cells that are created during meiosis, only one will be created. The other 3 will either get reabsorbed, become atretic, but they will not be chosen to be ovulated. With males, we know that all 4 cells will become viable sperm to allow for fertilization. So like mitosis, the stages in meiosis are basically named the same, and these stages and the events that occur in these stages are very familiar and basically the same as the stages in mitosis. So in Meiosis I, we'll have Prophase I, where we have the dissolution or dissolving of that nuclear membrane. We have the formation of our spindle apparatus. Metaphase I, we have the lining up of our chromosomes at that metaphase plate. Then anaphase I, we have separation of the homologous chromosomes to the different sides of the cell. So anaphase again stands, it starts with an A, think away. So separation of these homologous chromosomes separating away from each other. And then telophase, where we have a formation of a new nuclear membrane around our chromosomes, and finally cytokinesis with the separation of the cytoplasm. So prophase I, but we know that before prophase I, the DNA will be replicated in the S phase of interphase. Again, the nuclear membrane dissolves. We have the mitotic spindle starting to migrate. And in prophase I, this is when we have our first source of genetic variability, the crossing over. So non-sister chromatids will touch or synapse or forms a synapsis and exchange genes, creating a new combination of genes. So again, this is source number one of our genetic variability. The next phase in meiosis I is metaphase I. This is when we have our homologous pairs aligning in the middle at that metaphase plate. In this stage, we have our independent assortment where we have random lining up of the chromosomes. This helps to create gametes with different combinations of chromosomes from both mom and dad. And again, this is source number 2 of genetic variability. The next phase is anaphase I. This is when we have homologous pairs of chromosomes separating and moving away to the opposite poles. So the sister chromatids will remain attached at their centromere. Next is telophase I as well as cytokinesis. Again, think of telophase I sort of as the opposite of that first stage, prophase. So instead of dissolving the nuclear membrane, we have a formation of the nuclear membrane around the chromosomes. So at this point, the spindle fibers will disappear, and then with the division of the cytoplasm, we have cytokinesis, dividing the cytoplasm and the cell into 2 cells. So just a recap of meiosis I. Chromosome number has been cut in half, and the end result is 2 haploid cells. So we had a replication and then a division of those replicated chromosomes. So in sperm, eventually 4 haploid spermatocytes are made. And again, this is at the end of meiosis II. And again, in females, only 1 ovum is made, which is what is going to occur next. Sister chromatids get divided into daughter cells. This resembles mitotic division, however DNA does not replicate. And again, we have those 4 cells, 4 haploid cells with unreplicated chromosomes, and again producing 4 spermatocytes, and then 4 oocytes. However, only 1 ovum, 1 egg, will be released. So in meiosis II, we have the separation of our sister chromatids. So prophase II, we have again dissolving of that nuclear membrane. Metaphase II, we have the lining of the chromosomes at that metaphase plates. Anaphase, we have the moving away of those sister chromatids. And then finally telophase II and cytokinesis with the formation of 4 haploid daughter cells, and cytokinesis with the separation of the cytoplasm into those 4 cells. So this is exactly what happens in mitosis. So just a summary and the results of meiosis. We go from a diploid cell, And then after 2 rounds of meiosis, we end up with 4 haploid cells with 1 copy of each chromosome. So the end result, we have 4 genetically different daughter cells. And again, in females, only 1 haploid cell is chosen every month to be ovulated. Now let's talk about what happens when chromosomes don't separate normally. We have a situation called nondisjunction. Nondisjunction is what occurs when chromosomes fail to separate. So here we see our diploid parent cell, and instead of having in the first round of meiosis, or the end product of meiosis, 4 different cells with that are haploid in number, we have one that has 3 in this cell, and this cell only having 1. So this actually this produces what's known as a trisomy, and one of the more common trisomies is what occurs when there's an extra chromosome, specifically at chromosome 31, which occurs in Down's syndrome. So the most common error in meiosis occurs when the homologous chromosomes fail to separate. This is known as nondisjunction, or not coming apart. If nondisjunction occurs, then an abnormal number of chromosomes can find their way to the gametes, and a disorder of chromosome numbers may occur. A monosomy can occur if they are missing an entire chromosome, whereas a trisomy, where you have 3, can occur if they have an extra chromosome. So normally we know that each chromosome there should be 2. But here, mono, there's 1, and where there's missing an entire chromosome, and trisomy, there's an extra chromosome. So non-destruction of homologous chromosomes can actually occur in 2 different ways. First, we can have a normal separation in meiosis I, but abnormal separation in meiosis II. Another way that non-disjunction can occur is when we have abnormal separation in meiosis I, but then normal separation in meiosis II. So I'm sorry, Down syndrome actually occurs at the 21st chromosome, so trisomy 21, not 31. So Down syndrome, also known as trisomy 21, this actually can occur in 1 in 700 children that are born in the US. And we know that this risk or frequency will increase with maternal age, or the age of mom. So as we can see, as the age of the mother increases, this also increases the chances of her having a child with Down syndrome, or trisomy 21. So these are infants with Down syndrome per 1,000 births. And as we can see, once the mother starts reaching the age above the age of 35, the risk increases significantly. So trisomy 21, so we know at chromosome 21 there is an extra chromosome here, so there's 3, so tri means 3. And Down syndrome, people with Down syndrome do have a distinct physical appearance. There may be some mental retardation, as well as some underlying cardiovascular or heart problems. With regards to functioning, it is a range. You can have, highly functional individuals. They can, work, they can drive a car, they can do all sorts of things, versus, those that are not as functioning. So it's a huge range for patients with Down syndrome. Another chromosomal abnormality is known as Klinefelter Syndrome. This occurs in males that have an extra X chromosome at the sex chromosome location. So instead of being XY, they will be XXY. So males with Klinefelter syndrome are taller and have a less muscular body than males their age. There should be THEIR. They will have broader hips and longer legs. They have what's known as gynecomastia, which includes the appearance of larger breast. Weaker bones, a lower energy level, a smaller penis and testicles, so smaller reproductive organs, delay in puberty or have a small amount of puberty occurring, and then less facial and body hair following puberty. So because of this extra X chromosome, they actually have more female features, specifically gynecomastia. On the other hand, we have what's known as triple X syndrome. This is a female with an extra X chromosome at the sex chromosome location. Characteristics, they may appear normal, however have lower intelligence, are quiet and passive, may have a delay in speech, and may have delayed motor skills. Then we have a monosomy, so known as Turner syndrome. This is when the female is missing a copy of the X chromosome. So her sex chromosome on the karyotype is actually XO, so she's missing an extra copy or a copy of the X chromosome. So signs and symptoms of Turner syndrome. Possible symptoms in young infants include swollen hands and feet, a wide and webbed neck, as we can see here, and a low or indistinct hairline. As they age, in older females, some of the signs and symptoms include an absent or incomplete development at puberty, as well as a broad chest and widely spaced nipples. So this all occurring because there is a missing X chromosome at this location, as you can see in this karyotype. So an example of a monosomy. So these are some of the genetic disorders that can occur when we have non-disjunction of our chromosomes. And that is actually the end of chapter 7.